DNA transposons, class 2 transposable elements. Let us look at the learning outcomes of this session. So, DNA transposons can move from one locus to another locus of the genome by what is, in layman's term, known as the cut and paste mechanism. Now, in fact, cut and paste mechanism is a non-replicative mechanism of transposition. The DNA transposons, classified as class 2 type of transposable elements, have transposase and an enzyme that can enable excision of these elements and integration at another locus, which is generally referred to as the target site. Target site duplication is in fact a hallmark of DNA transposons. These transposons have been associated with genomic evolution, and due to its structure and function, these have been used as biological tools for transgenesis. Now let us actually understand the structure of DNA transposons. So the DNA transposons typically have a ORF of transposase. So they are able to give rise to the transposase enzyme, and the ORF per se is flanked on both the sides by what is called as the terminal inverted repeats. And it is these terminal inverted repeats that are able to function as sites of recombination. So class 2 transposable elements are, if they have the transposase, then they are considered to be autonomous transposable elements, which means that they can on their own cut or excise themselves out and integrate into a target site. That's the reason why they're called as autonomous. And it is because of the presence of transposase ORF that they are able to carry out this function. Now, what is interesting is that in many transposable elements, say for example in bacteria, there are, other than transposase ORF in the transposable element, other ORFs as well. For example, certain bacterial transposable elements also have genes or also have ORFs that gives rise to products that render antibiotic resistance. Now, as mentioned earlier, the terminal inverted repeats are actually the sites of recombination. So their integration is thanks to these inverted repeats, they enable recombination to happen. And on both sides of the terminal inverted repeats is what is called as a target site duplication. Now we would try to understand how these target site duplication sites are formed. But they are basically short, direct repeats. Now, when you look at inverted repeats, you can see that the sequence that is present this side, in this case, it will be present in the other side. So, both are, with respect to each other, inverted. Whereas the target site duplication are direct repeats, and they can be as short as 2 nucleotides or 9 nucleotides or 5 nucleotides likewise, but they are not very, very long. And they are formed as a consequence of recombination. Now, class 2 transposable elements, if they are without the transposase, then such transposable elements are called as non-autonomous class 2 transposable elements. So those DNA transposons that are non-autonomous do not have the ability to excise themselves and integrate into a target site. They cannot do it on their own, but they will have to be dependent on another autonomous class 2 type of transposable element. So they would have to use the transposase of the autonomous transposable element to carry out its excision and integration into a target site. For example, Barbara McClintock in her studies with Zia Mays, that is maize, was actually looking at the transposable elements AC and DS, of which DS was non-autonomous and AC was the autonomous element. and these were responsible for formation of the different kernels in the maize. Now let us look at what is the non-replicative mechanism, or how is the cut and paste mechanism happening. So as we can see over here, the transposable element is present in a DNA, and as you can see, it has a characteristic terminal inverted repeats, and within it would have the ORF for transposase if it is autonomous. Now, the transposon, this transposon is cut. So the DNA is cleaved in such a way that both the strands are cut. And so the transposase is actually responsible for cutting the DNA at the sites exactly close to the flanking inverted repeats. So as you can see over here, you have the complete transposable element being cut from the DNA that was the host DNA. So now what remains over here is a double-stranded break, and what remains over here is a double-stranded break which will have to be brought together, and therefore repair mechanism has to, a double-strand break repair mechanism or non-end joining homologous recombination or the double-strand break repair homologous recombination has to happen to ensure that the old DNA again joins each other. In the meantime, the excised transposon, because of having been cut at the ends, has a 3' OH formed, and this 3' OH will now act as a nucleophile. And we all know that nucleophiles are able to attack a phosphodiester bond, and that attack of the phosphodiester bond is at a target DNA. Now, interestingly, some of the transposable elements are not dependent on sequences to carry out the nucleophilic attacks. So basically, the transposable element is able to integrate anywhere randomly in the genome. Or you do have certain families or types of transposable elements which are able to integrate themselves only at preferred sites. So it may be sequence specific. Now, what is also very interesting is that when the 3' OH of the transposon that has been excised out cuts, it cuts in such a way that the target DNA is cut staggered. That means it is not cut exactly at the same position. It will not cut exactly at the same position. One cuts over here and the other cuts over here. So what you will have essentially is a staggered end. So this is the staggered end for this, and this is the staggered end for this. So this is how the cut generally happens. It is always a staggered cleavage at the recipient DNA. Now, what is important to note now is that you would have the transposon now through transesterification bind to the cut ends of the recipient DNA. So the nucleophilic attack is eventually leading to formation of a phosphodiester bond with the recipient DNA. However, at both the ends, because it's a staggered cut, you can see that this part of the DNA and this part of the DNA, there is gap. So what happens is that the DNA repair system will fill up these gaps. In the process of filling up the gap, the new DNA that is formed on the both ends is eventually the same site. So the sequence here and the sequence here is the same. And that's the reason why it is called as the target site duplication. So suppose you had TA over here, you would have the new DNA having AT. So if you had AT over here, because you have a TA over here, you would have a AT over here, and so the new DNA will have a TA. So basically TA, TA, AT, AT. So you effectively have the same sequence. It's a direct repeat that has formed at both the ends. of the transposon once it gets integrated into the recipient DNA. And that is why you will always find transposons flanking their inverted terminal inverted repeats. Flanking it, you would always have the target site duplication direct repeats. So therefore, this is essentially a hallmark of DNA transposons. Now, DNA transposons have been found to have a role to play in evolution for the simple reason that the DNA elements can actually cut themselves out from a part of the genome and integrate in another part of the genome. So therefore, when that happens, it can lead to loss of function or gain of function, or it can regulate gene expression differently once it has been transferred from one point to another point. So this constant ability of the transposable elements to be able to jump from one side to the other can lead to changes in the functions of genes. And because there are changes in the functions of genes, you could have the species evolving to become either better or to become stronger, etc. So therefore, you can see a lot of evolution that has happened because of DNA transposons. So you can see there are varied types of DNA transposons. TC1 Mariner transposons, you have the piggyback transposon, you have the P elements which are commonly found in Drosophila. So all these major types of DNA transposons have been found to be distributed across all the different supergroups that are present in the eukaryotic tree of life. And you can see that the dispersion or the presence of these DNA transposons are not same. They are different in different supergroups, and therefore you may be able to identify a family depending on what is the kind of, not identify, or rather you will be able to study the evolution pattern that has happened by actually comparing the presence of these different DNA transposons. So it's easy to understand that the ancestor of the eukaryotes already had, diversified to different supergroups. And that's why it is said that DNA transposons have a very important role to play in evolution. Interestingly, transposons can also be used as biological tools because of this ability to cut themselves and paste at another site. So therefore, it has had applications in genomics. And the major application has been in transgenesis, where a gene of interest can be inserted into a target cell. And also it has been used to carry out mutagenesis, and this in fact insertional mutagenesis, and this in fact has helped understand a lot of different pathways involved in various diseases or in embryogenic development, regulation of gene expression, and so on and so forth. Now let us understand how a commonly used DNA transposon system works. So in a DNA transposon system which is used for transgenesis, okay, you have one vector. So this is basically a vector, and you have the vector having having a transposon structure. So this is in fact what is called as a transposon vector. So as you can see, it has a typical terminal indirect repeats. You can see these two are the terminal indirect repeats, just like it is in a transposon. And at the center, there is an ORF which contains the gene of interest. So therefore, this transposon vector now contains a transposon element, but instead of this transposon element having the transposase ORF, it has the gene of interest. So if this has to be cut from here and transferred to a recipient site, this transposon vector will not be able to work on its own simply because it does not have the transposase. So this is not an autonomous transposable element. This is a non-transposable element. This is a non-autonomous transposable element. So therefore, this has to rely on transposase from another system. So therefore, along with the transposon vector, always is used what is called as a transposase expression vector, which would have a promoter, which would have the transposase gene with a poly A tail. So when you have the polymerase transcribing this, you would have the transposase mRNA being formed and that mRNA being translated to transposase and that transposase in the system. So in the transgenic cell in which both of these vectors are added, one is going to synthesize the transposase and that transposase is needed to cut this non-autonomous transposable element. So then when the transposase is formed, the transposase is able to insert this gene of interest into a target. And so as you can see, suppose this is the target DNA, then due to the integration, due to the transposase able to integrate, the gene of interest gets integrated into the target gene. And this is how you can transfer a gene of interest into a particular genome of choice. So this is why, or this is how you can have the DNA transposon system be used to carry out transgenesis. So let us make the conclusions. Okay, before going that, one of the very interesting DNA transposons is a sleeping beauty transposon that has been effectively used in different species, including humans. You can read more about the sleeping beauty DNA transposon system. The DNA transposons that are autonomous are elements having transposase ORF flanked by terminal inverted repeats, which are the sites of recombination. The transposases aid in cutting the transposable element and also integrating it either a random site or at a preferred site. And that is dependent on the type of transposon that is. As the integration involves cutting the target site and creating staggered ends, gaps remain after integration and these gaps are filled up to form what is called as a target duplication sites, which are therefore hallmarks of DNA transposon system. The DNA transposons have been identified and are diverse in their presence. So therefore, they are connected to evolution. Moreover, due to its ability to be cut and integrated, transposons are being used in genomics. So DNA transposons with its typical non-replicative mechanism, more commonly known as cut and paste, has found use as a biological tool to study genes, development, evolution, and diseases. Thank you.